Fiona has won many awards recently, most prominently won a Starter Award from the European Research Council, which is very highly sought after and extremely competitive. So she is doing really innovative work, and it's exciting to see that be acknowledged through this award. And after that, we will hear from Stefaan Verbruggen. Fiona's at University College Dublin as well. Stefan was at Sheffield University previously, and now he's at Queen Mary, and they were in the same PhD lab, but now he's doing similar work, but also a lot of computational work. So he's going to share some really interesting in vitro work, on a chip work, and also some wonderful work, especially around osteocyte measurement, doing computational modeling. And so it'll be really exciting to see the through line between both of our guests and also hear about their leading edge work. And so we will like to invite some questions throughout. So I will have a mic if anybody wants to ask a question spontaneously, because we do have some folks on Zoom. So we want to make sure they can engage, but we're really hoping for a vibrant discussion. And so join me in welcoming our guests.
Okay, well, that's a great start. Thanks very much, Kelsey, for the introduction, and thanks very much, everyone, for coming to our talks. So yeah, I'm going to give you a brief introduction into what my lab is now doing. And so what we're kind of trying to look into is employing bioengineering tools or technologies to try and develop novel therapeutics for the repair of damaged or diseased bone. And so how the talk will progress today is the first section is work that we did with developing novel bioinks and trying to deliver growth factors to repair large bone defects. Then I'm going to give a brief introduction into our osteosarcoma therapeutic that we came up with, which was gene delivery to try and regenerate osteosarcoma bone regeneration while also suppressing tumor growth. And then we'll hopefully get into some 3D disease models, which, as Kelsey described, is what my ERC is going to be on. And then hopefully I'll have time to briefly go into some immunoengineering work that we're doing now within the lab, where we're trying to target the immune system to try and actually kill the tumor itself, because as osteosarcoma is quite a cold or immune cold tumor. So the first thing we'll go into is biomaterial design and 3D printing. So I personally think bone is obviously the most superior tissue. It is constantly repairing itself due to normal everyday activities. And so when we were thinking about trying to repair large bone defects, we said we'd look to the tissue itself. And so when normal fracture happens, you get this formation of a hematoma, and then you get this fast influx of vascularization within those first few days. And there's been loads of studies that have shown that if you don't get this influx of vascularization in those first few days, you get impaired bone regeneration. So we were kind of looking at growth factors, and so we decided, okay, so we need to get this fast in growth of vessels. So can we deliver the growth factor VEGF gradient in a quick and timely manner to try and produce that influx of vascularization? And then over time, you get this formation of a bony callus, and then eventually the bone tissue will repair itself. So you have this slow, sustained release of what we were looking at, which is BMP2, and how can we slowly, sustainably release that to try and model or to try and regenerate the damaged tissue so that we eventually repair the tissue. So during my postdoc, I was in Danny Kelly's lab, and he was doing a lot of work with 3D bioprinting. And so we were using 3D bioprinting to create this implant for large bone defect repair. And so what we did is we created this scaffold or this PCL framework in which we were able to 3D print inside. And so the great thing about this framework was that it has lateral and horizontal porosity, so it allows for the bone tissue to form down through it, as well as that influx of vascularization. And then within this framework, we were able to deliver specific growth factors. And so we were looking at trying to develop our bioink, and we had done a lot of work with alginate. And so we used RGD gamma radiated antigen as our base bioink. And then to that, we were able to use different nanoparticles to try and tailor the release profile of the different growth factors. So the first one we were trying to develop was that vascular bioink. So we were trying to deliver that VEGF in a fast, sustained manner. So we added methyl cellulose just as a thickening agent. And then we added nanohydroxyapatite, as it was shown to quicker the release from the bioink over 10 days. So we got that nice fast release of the VEGF. So then we wanted to see what effect that was going to have with vascularization. And so vessels tend to move towards a VEGF gradient. So they like to move towards a higher level of VEGF gradient. So could we use 3D printing to create this gradient? So we had one group in which we put in our RGD gamma-rated allogeneic group within the whole thing with no VEGF. We had one group in which we delivered our VEGF Bioink, but also we delivered VEGF. Here there was a homogeneous delivery of the VEGF throughout the entire construct. And then finally we had a gradient group in which we delivered a high dose of VEGF in the center and then no VEGF around the outside. So we created this gradient with the hopes that we were going to increase the vascularization into the center. And so we subcutaneously implanted this in the back of mice. And what we found was by having that gradient of VEGF, so the total amount of VEGF in both constructs is the exact same, but by incorporating in that gradient using 3D printing, we were able to significantly increase vessel influx into the center of the construct, more so than just having a homogeneous VEGF gradient. So then we were like, we wanted to test this for bone repair. So we developed our vascular bioink, which has our alginate, methylcellulose, and nanohydroxyapatite. But then we wanted to slowly, sustainably release BMP2. And so we added in a nanoparticle known as lapinite, which basically electrostatically binds to the BMP2. So it really slowly, sustainably releases this BMP2 because of this electrostatic connection between the BMP2 and the lapinite. So we're able to create this bioink with a nice, slow, sustained release of BMP2. We cross-linked these and 3D printed them within our now nice framework. And so we created 3 groups. One, which was the same as what we had sub-Q, which had the VEGF gradient in the center, no BMP2 around the outside. The second was just the BMP2 gradient. So we had BMP2 around the periphery with no growth factor in the center. And then second one was our dual growth factor or dual gradient group, which has our BMP2 around the outside and our VEGF in the center. And so we implanted these into a large segmental defect model in a rat, and we wanted to see how the bone repair was happening. So if we just look at CT, we can see that even at 8 weeks, you can see a significant increase in bone volume in our gradient group, and this continued over 10 to 12 weeks. What was nice is that all of our growth factor gradient groups did significantly better than our MT, so even just having a VEGF gradient does increase the bone regeneration capabilities. When we looked at micro CT then as well, we could see that we are getting some bridging, but what was nice is having that PCL framework seemed to promote the bone through the PCL frameworks. We got no ectopic bone formation. The bone seems to go down through the PCL framework. And so we had very little ectopic bone formation. So then that was my first postdoc. And then I started writing my first Marie Curie grant. And so usually when I would start all of my postdoc presentations, I would say that large bone defects occur due to the fact that traumatic injury or tumor resection. And so that's delved me into the cancer field. I was like, well, I keep saying about tumor resection, but like, how do you regenerate the bone post-tumor resection? And so when you start talking to orthopedic surgeons and you suggest all these cool tissue engineering strategies that we've developed with growth factors, could we use them post-tumor resection? They're very anti any sort of growth factor. Because if you deliver any sort of growth factor there and there are any cancer cells, are you now going to promote cancer growth or even promote metastases by having that influx of vessels? So we kind of went back and we were trying to figure out what's the best thing. So the particular disease that I looked at for bone cancer was osteosarcoma. So osteosarcoma is a highly aggressive bone cancer that tends to affect children and adolescents and young adults. The current gold standard treatment for osteosarcoma patients is pretty aggressive. It's systemic chemotherapy, full tumor resection. And when you talk to orthopedic surgeons, they would rather remove the limb rather than leave any sort of margins. And they actually have shown that, like, if you do bone restructure, they actually have worse outcomes than if you do the full amputation. They then have a second round of chemotherapy. The 5-year survival rate for resistant forms of osteosarcoma is still below 20%, and there's been no change in treatment since the introduction of chemotherapy in the late 1970s. What I was interested in, there's also been limited research focusing on either bone salvation or bone regeneration post-tumor resection because of this, because it's such an aggressive disease. But the other thing is that the patients also have to undergo chemotherapy, and they've shown that when the patients are undergoing chemotherapy, the bone regeneration capabilities are completely diminished. So how can we regenerate the damaged bone post-tumor resection without causing tumor recurrence, but while the patient is also undergoing chemotherapy? And so this is where we kind of went to gene delivery. And so microRNAs have been identified as gene expression master regulators, and they constitute an attractive target for treating cancer. Specifically, I had isolated MIOR29B as I thought it might have this triple therapeutic role. So in other types of cancer, not osteosarcoma, they've shown that in those cancer cells, this particular gene is downregulated. And if you restore this gene in these cancer cells, you seem to induce apoptosis. They had never shown it for osteosarcoma up to this point. The other thing which was interesting is in osteoblasts, where this gene is normal levels, and you increase this gene expression, it actually promotes osteoblast differentiation and mineralization. So we thought it might also be able to aid in the bone regeneration capabilities. And then also it's been shown to be inhibit angiogenesis or anti-angiogenic, which when you're thinking about bone repair and you know that you have to have that influx of vascularization in order to induce bone repair, it's hard, but for cancer, when you're thinking about bone repair, if you have an influx of vascularization, you might promote metastases. And the 5-year survival rate for osteosarcoma patients when it develops to lung metastases goes from about 70% to 20%. So trying to deliver microRNAs have their own challenges. So you can't just deliver a microRNA on its own. It has poor penetration into the cell network. It's rapidly degraded. And if you just deliver it systemically, you can activate the immune system and have these off-target effects. Also, this particular microRNA is in a load of different cells. So trying to deliver it locally just to the tumor and bone cells was something that we were looking into. And so when I did my Marie Curie in MIT, the lab was working on a polymer-based nanoparticle known as PhEA. The great thing about this polymer is it's highly biocompatible due to its backbone of repeating ester groups that when it enters the cells degrade by hydrolysis. So they have really good loading efficiencies as well. So when you're creating it, it basically creates it around the microRNA. And then we did a lot of work comparing it to lipofectamine and showing it had a significant better uptake when you use this particular nanoparticle other than the commercially available lipofectamine. So we developed our nanoparticles. We created our microRNA nanoparticles. And the first thing we wanted to do was see what happens when we transfect the cells. So we used mesenchymal stromal cells as our pro-bone cells. And then we used osteosarcoma cells. And what we were looking to see is whether or not we got this pro-osteogenic, anti-angiogenic, and tumor suppressive role in these cancer cells. So if we first look at our mesenchymal stromal cells, the nanoparticles are stained pink. You can see the cells in the nuclei, and you can see that the nanoparticles do, in fact, go into the cells. And what was nice is we could see no cytotoxic effect due to the addition of these nanoparticles to the healthy mesenchymal stromal cells. But you can see this nice upregulation in ALP activity, so a marker for bone regeneration, and we see this inhibition of VEGF expression in the MSCs due to this microRNA. So then we looked at osteosarcoma cells, and again, we could see that the nanoparticles do in fact transfect the osteosarcoma cells, but what was interesting is here you can start to see the cytotoxic effect in these osteosarcoma cells, where we could see a decrease in DNA content. But I thought this might have been a mistake, so I went and did the study again, in which we looked at cells undergoing apoptosis by flow cytometry, and so these are the healthy cells that have been either treated with the microRNA, the scramble, or the chemotherapy. And you can see here, there's not a huge difference in the cells undergoing apoptosis in both the microRNA or the scramble group, whereas all the cells are undergoing apoptosis in the chemotherapy group. When we move on to our osteosarcoma cells, we can start to see this selective apoptosis with the microRNA group, where you see a slight increase in the cells undergoing apoptosis in our microRNA group compared to our scramble. So then we wanted to figure out how these cells are undergoing apoptosis. We looked at the apoptotic pathway, and so apoptotic pathway has two forms. There's extrinsic apoptosis, or there's extrinsic apoptosis. So we believed that it was extrinsic or intrinsic apoptosis, in which the nanoparticle is taken up by the cell, it releases its microRNA, and it seems to create this intrinsic apoptotic effect. So we looked at some genes for extrinsic apoptosis, and we can see that the microRNA, like chemotherapy, doesn't seem to promote the extrinsic apoptotic genes. But when we looked at the intrinsic ones, we can see, similar to chemotherapy, it seems to be downregulating BCL2 expression, as well as upregulating BAX expression. We saw this with PCR as well as Western blotting. So we're pretty excited. So we wanted to see therapeutically how this would work in an osteosarcoma model. So we developed a lung metastases orthotopic osteosarcoma model, in which we created a hole in the tibial plateau, we injected in the tumor cells, and we allowed the tumor to form for 2 weeks. Just before we applied any sort of therapeutic, we did CT analysis to see what effect the cancer was having on the bone tissue beforehand. And then we developed a novel hydrogel system to deliver the microRNAs locally to the tumor site as well as the bone cells so that we weren't getting any off-target effects. We compared it to systemic therapy of doxorubicin, and then we also did CT analysis 2 weeks post-treatment to see what effect it's having on the bone. Our delivery system was just a hyaluronic acid-based delivery system, which we were able to inject in situ, so a cross-linked right in situ, so there was no further operation to apply the hydrogel. And then when we fluorescently labeled the nanoparticles, we could see that due to the hydrogel, we were getting this nice localized delivery of the nanoparticles directly to the tumor site and no off-target signaling elsewhere. So what effect was this having on tumor volume? So you can see with our controls over the 15 days, the tumor volume seems to continuously increase. But the second we deliver our microRNA, either alongside doxorubicin or on its own, we might get this nice suppression in tumor volume in the animals. What effect did this have on survivability? So again, we could see that in our no treatment or dox treatment alone, there's not a great survivability for the animals. But the second we add in our microRNA or our microRNA plus dox, we get this nice increase in survivability. It's a pretty aggressive animal model. And so although we increase survivability, most of the animals actually ended up dying due to the presence of lung metastases. So we were kind of disappointed in the fact that we did all this nice work locally, but because it's such an aggressive cancer, that maybe we need to look at trying to deliver something systemically as well. What was interesting is then osteosarcoma is an osteolytic tumor. So in order to grow, it basically eats away at the bone tissue. So we can see here the percentage change in bone volume pre versus post-treatment. And you can see with the controls, they're getting a reduction in bone volume due to the growth of the tumor over time. But the second we add in our microRNA, we start to see this nice change in that and an increase in bone regeneration around the tumor. However, they did eventually die of lung metastases. So now we're trying to figure out what we can combine with this microRNA delivery system to try and help treat the metastases. Okay, so then I had this idea. So osteosarcoma mostly metastasized to the lung tissue. Unlike other cancers, which metastasize to loads of different tissues, for some reason, osteosarcoma predominantly metastasized to the lung tissue. So I believe that there's obviously some sort of crosstalk, you're welcome, Kelsey, between the bone tumor and the lung tissue that must be happening that primes the lung tissue for cancer cell engraftment. But this is something that's really difficult to do when you're looking at animal models, right? Because if we think about the metastatic cascade for osteosarcoma, you have this formation of the primary tumor, then you have the invasion of the cancer cell within the vascular network, and then the extravasation of the cancer cell from the vascular network into the lung tissue. But in an animal model, that's really hard to decouple how these two tissues are actually integrating because there's so many other things happening all at the same time. So could we develop an in vitro model of the disease where we could decouple this and figure out how these two tissues communicate? And if so, if we could figure out how they communicate, can we figure out maybe novel targets to try and treat it therapeutically to stop lung metastases? So the first thing we tried to do is try to model the primary tumor. So we're doing this in two ways. The first way that we did is we created a co-culture spheroid model of osteosarcoma, which had mesenchymal stromal cells and osteosarcoma cells. And when we manipulate the ratio of osteosarcoma cells to mesenchymal stromal cells within the spheroid model, we were able to model either early or late stage osteosarcoma by the increase in the DNA content, so the cells are continuously growing when there's more osteosarcoma cells, the spheroids get much bigger, and then we get this increase in prognostic genes, MMP9 and beta-catenin, which shows that we're kind of increasing this growth of cancer within it. So we kind of had our primary tumor, and then we wanted to model vascularization. And so when we were thinking about trying to model vascularization, we kind of went into the organ-on-chip field. So an organ-on-a-chip is a device, I'm sure that you all know, it's pretty much the size of a USB key. It can be made out of usually a soft polymer, and you can model certain aspects of organs to try and understand how they grow and develop and maybe react to different therapeutics. The organ-on-chip technology can offer insights into the inner workings of the effects of drugs just before you have to go to a full clinical trial. Also, the great thing is that the US Congress approved the FHEA Modernization Act 2.0, which means you can now use organ-on-a-chip data for drug discovery instead of animal models, this is a huge thing for the field. The great thing for osteosarcoma patients is that it's a pretty rare disease. So trying to develop clinical trials for osteosarcoma patients is really difficult to get even enough patients to run full clinical trials. So if we could use patient tumor samples within our organ-on-a-chip device, we could create multiple chips where you could run multiple different drug distributions and figure out which is the best one for that patient, or even create mini clinical trials on chips where you're getting tumor samples from multiple different patients in one place, and you can run the same drug and see how it goes over a huge population, which hopefully would mean that you could theoretically streamline the clinical trial process, and then means that maybe the drugs that do hit the clinical trials have a much better option. So when we decided this, we looked at trying to create an organ-on-chip. So this particular organ-on-chip that we were looking at just had a central channel in which we filled with fibrin as our hydrogel, and then we also included our spheroid model of early and late stage osteosarcoma, and then we had endothelial cells. And so when we put these all in the center channel of the organ-on-a-chip, we left it for a couple of weeks and saw what happened with the tumor spheroids and the vascular cells. And so what you can see at 24, 3, and 5 days, the vascular cells are stained here in green. The white circle is the spheroid. At 24 hours, the cells are kind of roundish, but over the 5 days, the vascular cells seem to be completely repelled by the early spheroid model. They seem to not like it. They form things around it, but nothing close to it. However, when we put in our late stage osteosarcoma spheroids, you can see that the vascular cells seem to grow towards it and eventually completely engulf the spheroid. And so you can see it a bit better with a zoomed in version. So again, early spheroids, you can see no green cells around it, just the spheroids on their own. The second we incorporate in the late stage, you can see that the HUVECs seem to engulf in the tumor spheroid. We then also took the media from this to figure out maybe what was going on, and what we could see is that we see an increase in IL-6 and IL-8 expression in the late stage versus the early stage, and we also get a significant increase in VEGF. And these are all markers of metastases. So there is some sort of cues that are coming off this osteosarcoma that might promote metastases. So the final stage is then trying to look at lung engraftment. And so for this, I was lucky enough to know Stefaan, who put me in contact with Queen Mary. And so we did like a small proof of concept grant with Emulate Bio, in which they have created this lung chip device. And so within this lung chip device, you have your epithelial layer and your endothelial layer, and then in between you have a membrane. So you can see the two cells there. The great thing about it is that you can flow air along the epithelial layer to mimic breathing. You can also incorporate in stretch, so it also mimics the biomechanical aspects of breathing. And so what we wanted to see is whether or not we can model that pre-metastatic niche, those changes that occur when you take the conditioned media from our osteosarcoma patients and put it on the lung chip. And so we took our conditioned media and we just loaded it and flowed it through their lung chip device. And what we were trying to see is whether we were getting this pre-metastatic niche or this formation of a leaky vascular network and maybe ECM remodeling in the epithelial cells. So the first thing we did is we looked at the permeability from the vascular layer into the epithelial layer. And so what we can see is this increase in permeability when we load in our conditioned media, and we can see the formation of these holes. So before we have the conditioned media, this is a perfectly flat, like no gaps between the cells. The second we add in our conditioned media, we get the formation of these holes within the vascular network. And then when we took the epithelial cells and did some PCR, we could see an upregulation of collagen type 4 and a trend towards upregulation of fibronectin. So we are starting to get to see that epithelial ECM remodeling, or the cells becoming stickier for cancer cell engraftment. So my URC, which is on that, is trying to develop this model further. And so trying to create an osteosarcoma chip that also incorporates in the bone microenvironment. It's also going to try and incorporate in other types of cells like immune cells, and then trying to really pinpoint how these two tissues communicate. So my final bit is on immunoengineering. And so I was saying yesterday that the more I know about the immune system, the more I realize I know nothing. Specifically when it comes to osteosarcoma, there's very little out there. So, you know, for all other types of cancer, there's a huge push towards using immunotherapies to treat them. For osteosarcoma, there's very little out there. So we were kind of interested specifically in the tumor-associated macrophages. And so macrophages can form in either your M0 state, your M1, or your M2. When it comes to bone and bone regeneration, you obviously want an M2 phenotype, so you're promoting that bone regeneration, and less of an M1 phenotype. What's interesting in cancer is you want the complete opposite. So an M2 phenotype is pro-cancer growth. It basically promotes an anti-inflammatory response, so the immune cells don't come in and try and kill the cancer. Whereas if you have an M1 phenotype, the cells are much more likely to come in and try and attack the tumor. But at the moment, there was very little known on how osteosarcoma manipulates these two cells. And if they're already polarized towards one way, can they manipulate it towards the other? So I had a really good undergrad who this results came as I was doing them on the plane, as she sent them to me just before I got on the plane. So we took conditioned media from osteosarcoma cells and we put them on macrophages that have been polarized either towards an M0, an M1, or an M2 state. And so the first thing we looked at was DNA content, and we could see there's no real increase in proliferation by adding on the osteosarcoma cells. But when we look at IL-6 and IL-8 expression, we can see that by adding the conditioned media onto an M1 or an M2 macrophage, you actually are promoting that M2 phenotype. So we see a reduction in IL-6 and a reduction in IL-8 compared to the macrophages that had no conditioned media. What was interesting and actually was good to see Stefan's talk yesterday is the second then we looked at a transwell insert, so direct co-culture. So we had our cancer cells and our macrophages that have been polarized towards an M1 or M2. We saw some interesting results. So the first thing we saw is that if it's already an M2 phenotype and we culture them with osteosarcoma cells, you get a significant increase in proliferation of M2 macrophages. Not the same when you have an M1 phenotype. So it seems to promote M2 growth. But then when we looked at IL-6 and IL-8 expression, we can see the opposite. So it seems to promote the expression of IL-6 and promotes the expression of IL-8, which is counterintuitive because we saw the complete opposite when we took conditioned media. So there seems to be this crosstalk between the macrophages and the osteosarcoma cells. And it seems to, but then also IL-6 and IL-8 have been shown to induce metastases. So maybe they're promoting this expression that is then inducing the metastases in the osteosarcoma cells. So you can.
Can people hear me? Okay, great. So I love this concept and the way that you're modeling it, and I have to stop you since no one did, so I'll take a liberty. I think the really fascinating thing about IL-6 is that it seems to do different things in different tissues, and it seems to do different things in autocrine and paracrine fashion. So I think, like, if we think about IL-6 in the context of metabolic syndrome, for example, it's something that's likely made by either fat or the liver, secreted somewhere with downstream effects. But in muscle, for example, IL-6 is a really key reparative strategy. And I know it's probably too much undertaking and not central to the hypotheses here, but I'm wondering if you can try to separate those autocrine and paracrine actions by using this setup that you have to get us to understand a little bit more, like especially when you're thinking about local targeting versus systemic targeting. And are you thinking about that, or is there a way to do that using some of these tools that you've developed?
Yeah, so we're hoping to, the more you understand it, the more you're like, okay, now I want to knock some stuff down and I want to figure out exactly who's expressing this, right? Because theoretically, osteosarcoma cells express IL-6 and IL-8. So it's who's expressing this. Are they expressing it more than the macrophages, and how can we figure that out? The other thing is that IL-6 and IL-8 are also known markers for metastases. So although they are theoretically an M1 phenotype, they also might be being produced so that the osteosarcoma cells are going to go, okay, cool, it's being produced, I should move. So actually we looked at that. And so we basically did, within the transwell, we were able to show that if you just have osteosarcoma cells on their own and you looked at the bottom layer of the transwell, you can see that the osteosarcoma cells on their own didn't really move much. But the second we put in any sort of macrophages, they started to move a bit more. Now, we didn't see huge differences between what kind of macrophages. It just seems by putting them in with macrophages, the osteosarcoma cells seem to react differently. But yeah, we really want to figure out like who's producing it, how it's being produced, and maybe there's some sort of target there that we could look at. So I suppose my concluding remarks is as engineers, we have this amazing toolbox, right? So we have all these cool techniques that we can use to really try and answer some really cool scientific questions. And so I hope I've shown you that with our engineers toolbox, we could really develop these novel in vitro models of the bone tissue microenvironment in both the healthy and disease state. We could develop nice biomimetic biomaterials as a therapeutic and delivery system. And then also we can try and develop these nice ex vivo testing platforms for drug discovery to try and streamline the clinical trial process. So with that, I'd like to thank my lab and my collaborators for a lot of this work was done in postdoc when I was in MIT and as well in Trinity College. But most importantly, I have a lot of open positions. So any trainees who are out there who are super interested in to see how this disease works and trying to understand it, I have 2 PhD positions on the ERC, so on the meta chip, and so on developing the osteosarcoma on a chip device. I have a PhD position, which is looking at trying to incorporate other microRNAs as a therapeutic for trying to work on that therapeutic response, so delivering different, using the same nanoparticle to deliver different microRNAs. And then also I have a postdoc, so it's about 2 to 4 years working on the ERC's grant. With that, I'd like to think any more questions.
Wow, that was fabulous. I'm sure there's lots of questions. Does anybody want to start?
That was fantastic. Thank you. I have a quick question about the microRNA delivery. So I guess one of the things that you mentioned that struck me is that you're able to get better survival in the mice, but they all eventually die because the disease is so aggressive. And I was wondering if one could test this in perhaps a model in which you resect the tumor and you instead use it to treat residual disease, or perhaps use it also in like early stage osteosarcoma.
So absolutely. So weirdly, there were, up until my paper, there were any paper that was looking at bone regeneration post-tumor resection looked at 2 different animal models. So they looked at a tumor model in which they show subcutaneously that their therapy can shrink the tumor. And then they looked in a healthy mouse, like segmental defect model, how it regenerates the bone. So I was like, that's not indicative of what happens, right? Because the patients are undergoing chemotherapy, they've been in the disease state, so their repair capabilities is completely different. Originally, we wanted to do a resection model, but by the time the tumor grows to a size that's suitable, if we removed it in the tibia plateau, they would have very little limbs left. So we kind of just looked to see what would happen locally. I really want to develop a resection model, and I wonder whether in mice the best thing might be to do it cranially and form the tumor, and then you're able to resect it around the skull and then look at trying to regenerate it. I'm really interested in trying to figure out what's the best way of modeling that, or whether it's that we go to a larger animal model. So unless we go to a rat and then you have a bit more to play with. It was just, it's a tricky one to try and get a one that actually works. And then we're also trying to look at, we only did 1 delivery of the microRNA, and you could see that it's delivered over 15 days. But maybe if we delivered it a second time, and we might be able to see better results that way.
Great, thank you.
No problems.
Question?
This is just a strategy question. In thinking about trying to get drug delivery to work, and you used microRNA as your platform of choice, I guess. Was it more of a platform choice because microRNAs were more tractable? Was it because that was had a biological activity you're most interested in, or why didn't you choose something like an AAV or a protein delivery or a genome editing type of strategy? I'm wondering what drove that decision.
So originally we actually looked at trying to deliver BMP2, and I don't have that in it, because we saw with our in vitro model, when you deliver BMP2, you don't actually get an increase in cancer growth. So we were like, maybe we could deliver BMP2 and it wouldn't happen. So we delivered BMP2 using the same hydrogel delivery system, and we didn't see an increase in tumor growth. But the second you apply systemic chemotherapy alongside the BMP2, the biological activity of the BMP2 was completely diminished. So we were like, okay, so we can't use growth factors. They don't work at all when you deliver systemic chemotherapy. So we kind of went with the microRNAs because we knew it could be delivered. It was around COVID, so they were everywhere. And I pinpointed this particular one because it kind of had this triple therapeutic role that it could work. And I got really lucky that it worked because I had a limited amount of time. But now we're trying to figure out maybe there's better options out there. But that was kind of, it had this novel triple therapeutic role, and it wasn't hugely cytotoxic, and we hoped that it would work alongside a chemotherapy, which we showed it did.
Did you see what percentage of cells were able to take up or be affected by the microRNA?
Good question. I wish we did. I was trying to, yeah, no, is the honest answer to that question. We only really could see, we would have liked to kill the mice at certain time points and then do flow cytometry and figure out which cells had actually uptaked the microRNA. Just ran out of a lot of time. So yeah, my 2-year postdoc that was in the States because of COVID, I lost about a year, well, 8 months. So it was kind of what we could do with that limited amount of time. However, we're now currently bringing the model to Ireland. That's the plan, is to try and figure out what cells are uptaking it, whether or not we could deliver a second microRNA that maybe incorporates in the immune system, or could we deliver an immunotherapy alongside of the systemically.
Other questions? I can ask one more. Oh, Gertrude.
It's okay. I think I'll just talk out loud.
Well, there's people on Zoom, so then she'll have to repeat your question. So you might pass them.
No problem.
Hi, amazing talk. My question is regarding your organoid on a chip model, and you're looking at crosstalk between the two tissue types. So a lot of research has been done about the priming, how the osteosarcoma cells prime the lung niche with exosomes and everything. So are you also going to be looking at that, or circulating tumor cells maybe?
So yes, 100%. Yeah. So there's been loads of work with exosomes that have shown TGF-beta within the exosome seems to promote primate. So the plan is to hopefully develop a nice model with, and hopefully with the vascularization. So we were able to show that the vessels that form within the organ-on chip actually have lumen, and so you could flow things through it. So we're hoping that means that the cancer cells might move in it, and then we'll collect them and put them straight in. And the whole sort of the plan would be, if we could collect enough of them, we might even do some single cell sequencing as to what is different between this cancer cell and the cancer cells that still remains in the chip, and whether there's specific genes that make a cancer cell want to leave and go somewhere else.
Yeah, that was my other question too. The vessels that you see right now, are do they have lumen, or are they?
They do, yes. We flow dextran through it, and we can see that the dextran goes through the vascular network. Yeah.
Awesome.
And then the hope is to also incorporate fibroblasts to try and get better vessels and nicer vessels. Yeah.
Awesome. Thank you.
No props.
You might have time for one more.
Really fascinating talk. Thank you. I had a question about sort of the duration of the effect of the microRNA type therapy, like over what period of time, generally speaking, and maybe this varies by tissue type and a lot of things, but for what period of time do those sort of seem to be working? And then is there a period where they're sort of depleted and no longer effective or no longer acting?
So what we were able to show was that using the hydrogel delivery system, we could still see nanoparticles 15 days later, but after that, the fluorescent imaging showed that there weren't any nanoparticles left. So seemingly, as the hydrogel system degrades, it releases nanoparticles. Nanoparticles being taken up, they have their therapeutic effect. And it seems like anything post 15 days, we were getting very little signal. So the thing is whether or not we deliver it. At that time, we were like, let's just deliver it and see what happens. So now we're looking at, do we deliver it twice? Do we deliver a high dose, then a low dose? Do we deliver it, a multiple different timeframes? Because obviously it did have a therapeutic response in the 15 days, but that wasn't enough. It's a pretty aggressive tumor.
So generally speaking, like this therapeutic approach would be relevant to sort of like acute tumor effects as opposed to like longer term chronic sort of.
Yeah, I think the therapeutic in the clinic, it would be delivered once they resect the tumor. You inject in this hydrogel that has the nanoparticles, and it will mostly kill any of the residing ones and help in the regeneration. We haven't developed an animal model to prove that point yet, but that theoretically would be the plan, because they'll never not resect the tumor. So they'll always want to get rid of as much tumor as possible. So it's more, can we deliver something that will be delivered alongside the chemotherapy to help the regeneration capabilities of these patients so they don't eventually end up having to undergo amputation.
Right. Join me in thanking Fiona again. So as we transition between speakers, hopefully it's really apparent, and I think will become apparent again in Stefan's talk about how
phenomenologically, we're thinking about a lot of the same things. Like you might not spend a lot of time thinking about osteosarcoma. You might not spend a lot of time thinking about, well, everyone spends a lot of time thinking about osteocytes, obviously, but I mean, there's other, but a lot of these principles and approaches and solutions that people are looking for and successfully finding are things that can be applied readily to what we're doing. And so I think hopefully it's really energizing to think about crosstalk as a lifestyle and not as a disease specific idea, which of course was socially engineered by Dr. Allison. And I think that hopefully this will get you excited about, you know, going to talks sometimes that are in different areas that you might not think about. So I'll shut up now.
Thanks, Kelsey. So yes, well, I feel like I'm in a safe space, so I'm obsessed with osteocytes. Yes, I've sort of drifted. I mean, myself and Fiona, we were PhD students in the same lab, so we trained together. And we both kind of trained in bone mechanobiology and bone regeneration, and then we've sort of drifted more towards cancer. And she's nicely introduced basically everything for me, including organ on a chip work and cancer in general, so I don't have to do anything. So I'll jump straight in to say that I'm focusing a bit more on metastatic cancer, so breast and prostate metastatic cancers in particular. And the reason I'm focused on those is while osteosarcoma is devastating, it is just, as Fiona nicely said, it's a really rare cancer. Whereas patients who have late stage metastatic breast or prostate cancer, a lot of the pain and suffering that they're going through, the problem is the bone mets, the metastatic lesions in their bones. And really, at the moment, what we basically do is we throw osteoporosis drugs at the patients and hope for the best. That's, you know, it's more, there's more than that, but that is, we haven't really developed any, you know, therapies that target the tumor specifically, even though there's lots of work going on looking at it. So what happens? Well, you get your breast and prostate cancer cells that have spread around the body. They pop out from the lymphatic system or the vasculature into the bone marrow space. And then they start to give off growth factors, cytokines, and so on that kind of hijack the behavior of the osteoclasts and osteoblasts. They increase osteoclastogenesis, or if it's an osteoblastic tumor, they might increase osteoblastic behavior. You get a hard bony tumor. And really why these two cancer types, which are the most prevalent in either sex, like to go to bone is actually still a bit of a mystery. But none of those cells in that video are osteocytes, you might have noticed. And I think one of the reasons is when people think of, especially oncologists, when they think of the cancer cells arriving in the bone marrow, they think like, what do they meet first? Well, they meet osteoclasts and osteoblasts and MSCs. So we know a little bit about, there's some really good theories that have been well proven that cancer cells can influence osteoclastic behavior, osteoblastic behavior. A lot less is known on what these bone cells actually do to the cancer cells, like the crosstalk back in the other direction is kind of a big gray area. And very few people have looked into osteocytes, which I naturally think is weird because they're the most ubiquitous bone cell. They make up more than 90% of our bone cells. They usually control the behavior of the other cells in response to mechanical loading. They're kind of like a master cell. And they're overlooked in metastatic disease, which I think is a bit odd because I'm probably giving too much agency to the cancer cells. But if you're trying to control something, wouldn't you go for the foreman on the building site rather than the guys in the building, the guys laying the bricks or knocking the bricks? You'd go straight to the guy, to the person in charge. So I just kind of thought it was strange that it hadn't been looked at. However, when doing a bit of reading recently, I noticed that people have actually known that osteocytes are seriously disrupted in cancer since as far back as the 70s. There's this really nice paper called The Ailing Mythical Osteocyte that I'm just drawn to this paper for a few reasons. One, that name, that title. It was just written by a doctor in Texas in the 70s, 1975, whose name is Jay Stinson, and my husband's name is Jay Stenson, so it's a bit weird. I'm hoping it's not like him from beyond the grave or something. But just look at these images. So just for the people online, I'll use the mouse. So you can see here, this is you can't see, well, a wave. So this is a classic conversion system. The osteocytes should look like the ones on the left-hand side, all kind of around, organized concentrically around a nice blood vessel. They look kind of long and thin, and they've got these long, thin dendrites, kind of caniculi that reach out to each other. Don't need to tell most of the people here. And then next to it is this tumor region, this osteoblastic tumor. So I think this is osteosarcoma. And what this guy saw was that and what he noted was that the canaliculi were thick and blunted, and they don't look kind of long and thin. And it just looks, even just if you kind of glance at it, you can see those osteocytes look weird. And he looked at the more detail. He found areas where there was loads of canaliculi, loads of long, thin ones, but like basically no lacunae, like no osteocytes. And then he found other regions where there was like little evidence, like just here kind of shades of what might have been osteocytes, but are now kind of hypermineralized, and no real evidence of connections between them. And then he found what he called quite elegantly skeletons of osteocytes. So like a bit meta, but kind of the remnants of kind of damaged canalicular networks. And this is using just silver nitrate staining, which we kind of still use today. So it's amazing that far back, he even inferred, he guessed that there might be unusual electrical influences that might be causing these changes, chemical aberrations in a hypothetical conductive fluid, which kind of obviously we know now is important, but back then he didn't. And then that there might be mechanical disruption. And I like the way he finished the paper. He said that just like people, it would seem that whatever osteocytes are or are not, they've got problems too. And I just was kind of blown away that it's that far, like 50 years ago. People were already thinking about this stuff, and he basically was right. He should have maybe gotten a prize for this. Now, almost nothing has been done to look at osteocytes in that much detail using histology since then, in cancer, I should say. But there was a really nice recent study that looked at a metastatic breast tumor model. And I'll have to wave here now. So you've got your bone marrow here, so lots of fat cells. That's why they're all showing staining strongly in confocal microscopy. And then anywhere where it says TC, that's the tumor. These are successive slices within a confocal stack. These are all tumor cells. And then the little arrows point out the osteocytes. And what they showed was that you actually do have direct contact between the osteocytes and the cancer cells. So it's actually a bit hard to see here, but might be able to see that there are dendrites connecting the osteocytes to the tumor here, down here, here as well. So there's direct contact. But then they found that in further away from the tumor, using silver nitrate staining again, in the control bone, there's kind of lots of nice connections between the cells, but then the network looks kind of messed up further away. So it implies that there's so the cancer is doing something to the way the osteocytes organize their network. And then there was a recent study by Linda Bonewall's group that found that even non-bone metastases can broadly affect the structure of bone. So she looked at I can't remember off the top of my head, but they were let's say a lung tumor that's metastasized to the brain, or a Ewing sarcoma that's metastasized to some other part of the body. And even though there's no evidence at all and there's no tropism for bone in those models, there was still degradation of the bone tissue that wasn't explicable by any other. So it seems to be systemic, and maybe there's crosstalk. So that's for Kelsey there. I said those. So that led my lab to posit that osteocytes might actually play a direct regulatory role, either as a relay cell or possibly actually just a direct role in regulating metastatic cancer, which of course is a bit of an out there statement, but walk with me. So during my fellowship when I was in Colombia with Chris Jacobs, I kind of was trying to work out how to do this. And a bit like with what Fiona was saying, and I have post-traumatic stress from this, I decided to try and do a model of early metastases and established metastases. And I was thinking, okay, if you buy into my theory and you have a handful of cancer cells that arrive into the bone marrow, they're arriving to an environment that's largely regulated by the osteocytes. Whereas later on, if you've got a big tumor that's growing, there's probably crosstalk because you'll have enough cancer cells that they'll be giving off enough signals that they'll be affecting the osteocytes in return. So I kind of thought, well, that's basically one is a condition media model and one is a co-culture model. So that's what I decided to do. As my first experiment, first time working in the lab, as Kelsey alluded to, I was a computer nerd and I'd done some in vivo work, but not any in vitro work. And I thought, sure, how hard could this be? So I took conditioned media from osteocytes, added it on top of cancer cells, and then did a co-culture transwell experiment as well. So here's my lovely graph from my very first experiment, when I was fully convinced that I'd see a change, and I did, which is nice. And what I saw was that if you add conditioned media from, oh, apologies now because the color's not showing, but yeah, the order is the same. So on mine, it's like a lighter gray. But the conditioned media from the osteocytes in the middle bar decreased the proliferation of 4 different, 2 breast and 2 prostate cancer cell lines. So I'm only showing one here because it would be death by bar chart otherwise. And they kind of nudged them down a more migratory phenotype. And we measured migration using scratch wound assays and transwell invasion. So it seems like the osteocytes were tamping down the kind of proliferative behavior of the cancer cells and making them be kind of more go than grow, if that makes sense. So naturally, I thought that the co-culture model would be more exacerbated, and I found like it was the same basically as the control. So I was fully convinced that I had like messed this up. So repeated it. And it was just the same pattern. So that just like what Fiona's now going through. And she asked me yesterday, she was like, what should I do? And I was like, run away now. Because to start to pick this apart, the mechanism, you kind of really have to repeat this experiment basically 100 times. So it just implied to me that maybe there's something, if the osteocytes can suppress the cancer cells, then they can't do it in co-culture because something's shutting off their ability to do it. So there's some feedback. So I asked myself, what do the cancer cells produce? And one thing that all cancer cells produce lots of is TGF-beta. And we checked that using ELISA. We looked at other ones too, but TGF-beta stood out. And sure enough, yeah, there was a huge production of TGF-beta by the cancer cells. So I thought, right, well, if that's the culprit, then if I block the ability of the osteocytes to sense TGF-beta in the co-culture model, the behavior should be similar to the conditioned media experiment, because if that's what's causing this in the co-culture model. So I had a control co-culture and then a scrambled siRNA knockdown of the TGF-beta receptors on just the osteocytes. And sure enough, when I knocked down the TGF-beta receptor, you saw a decrease in the proliferation and an increase in the migration. So basically, that made the co-culture result look like the conditioned media result. So I was like, okay, right, the cancer cells are telling the osteocytes to do something, and they're doing it via TGF-beta. But what does TGF-beta do? Now, I mentioned that I was in Chris Jacobs' lab, and the other lab that and he's now a colleague of mine, Martin Knight. He's a cartilage person in Queen Mary in London. He is all the two of them. You know, you don't get far in their labs without doing something to do with the primary cilium. They're big fans, and I guess now I am too. I've joined the church. So what is the primary cilium? For anyone who isn't familiar with it, it's a slender organelle that protrudes from the surface of pretty much every mammalian cell. It acts as an important chemo and sometimes mechano signaling nexus. Controls really important pathways like hedgehog and Wnt signaling. The way it gets built basically at different stages during the cell cycle, and if a cell goes to divide, they disassemble it again. The way they do that is using IFT88, which is a little protein that clips onto the top of a motor protein, like a little backpack, and creates like a carriage to trundle up and down the length of the axoneme. And just like a Jenga tower, they basically grab little bits of microtubule and build it up. And then when the cell wants to divide, they just disassemble it again. And that also means that you can use this intraflagellar transport protein to shuttle things on and off the cilium. So a lot of TGF-beta signaling happens, smooth, frizzled, all that stuff happens on the cilium. It forms this kind of unique microdomain within the cell. So there's lots of biochemistry happening specifically within that microdomain. And that means that there's actually a structure-function relationship. So if you have a longer primary cilium, you can do more chemistry. So you can have more hedgehog signaling. If you have a shorter one, you'll see that there's just less. I don't know if it's just that there's less space, but in my head, that makes sense, that there's less space to do the chemistry. So you'll have a less of a hedgehog signaling response to whatever stimulant you're giving the cell. And you can see it here in super res microscopy and then just kind of on cells in vitro. Why have I given you that talk on primary cilia? Well, I knew from work in my own lab and in others that if you give TGF-beta to similar cells from the mesenchymal lineage, chondrocytes and osteoblasts, it tends to reduce the expression of primary cilia. So you see a knockdown in chondrocytes, and then in these pictures you can see the osteoblasts. You've got this is sort of what they look like, little long kind of little green things, and they sort of disappear. And you still have lots of microtubule staining there, but it's all in the cell cytoskeleton rather than focused around the cilium. So we figured, yeah, it'll probably have the same effect in osteocytes. And sure enough, it does. So these are MLO-Y4 osteocytes, the osteocyte cell line that I was using. And on the left-hand side, you can see, again, I'm just showing you one of the cancer cell lines, but it's just the left-hand panel is just the osteocytes on their own. The second panel is conditioned media from added to them from the breast cancer cells. And you can see that you have way less cilia indicated by the arrows, and the ones that you do find are quite truncated. They're quite small, like the figure in the inset. And you can block this if you have a TGF-beta receptor inhibitor, a small molecule inhibitor in the media with the conditioned media. You kind of get a bounce back, and I have all the data for this as well, but I don't want to kill you with bar charts. And you can do the same with an siRNA knockdown. If you knock down the TGF-beta receptor, you see like lots of cilia. They're still not quite as long as in the control condition, but they're still there. So that made me think, right, the cancer cells are doing something to the cilia, and it's via, I did want to check that it was, because there's other ways that the cilia can be constructed. So I wanted to double check that it was via IFT88, the specific protein, and we did PCR and showed that TGF-beta and the cancer cell conditioned media reduced the expression of that protein. So it is this IFT88 pathway that we're affecting. Okay, so if the cancer cells are knocking down, yeah.
But Tom said he might not need it because I think they were okay.
Okay, great.
My question is there also a difference in the acetylated tubulin? Are there other mechanisms in addition to IFT88 that you think are?
So I measured it's kind of difficult, but you can do it. But I measured the amount of microtubulin staining in the cells, and it wasn't like there was less microtubules. Like there was still, I mean, I don't have it in that for my own data, but in this paper, like you can see, there's still lots of microtubules there. There's still lots of tubulin. It's just not acetylated. It's just kind of floating around. So it's not a lack thereof. It's that they just aren't constructing the cilia. Of course, there could be some other pathway. It's just that we settled on this one, and I was running out of time and money, and yeah. So yeah, yeah.
So I mean, I have a related question because I should know more about the primary cilia, but is this a remodeling sort of organelle? Is it something, or are these cells just sort of differentiated without it because of the exposure to TGF-beta?
Basically the cells can continue to function. Like it's not like knocking one out. Knocking out the primary cilia completely, like with the CRISPR, is embryonically lethal if you do a global knockout. We do have some DMP1 Cres that are constitutive, like osteocyte linked. But it's the reason it was thought to be vestigial is we knew it was important for forming an embryo. It seems to be really important for patterning during development. So maybe the cells use it to know which way is up, I guess, and how to build what next to what. But when you lose the expression, you do lose things like, I don't have the data here, but we looked at COX-2, like mechanosensitivity of these cells after treatment with conditioned media or TGF-beta. And yeah, they lose their mechanosensitivity. So like the COX-2 expression in response to shear stress goes down. So I can't say if the cells are changing their phenotype. There could be phenotypic drift, or not drift, but phenotypic change. But these changes are quite short-lived, and the experiments are not long. So I don't think it's enough for the osteocytes to do something massively different, but it certainly affects the mechanobiological parameters that I was looking at. So yeah, so they didn't like it. I'll put it that way. Right. But I was asking the question, right? Well, if the osteocytes don't have primary cilia, can they still do what we saw them do in the first experiment? So if they're suppressing proliferation, if the osteocytes are suppressing proliferation, do they need the primary cilium to do it? Because we now know that the cancer cells are knocking down the primary cilia in the co-culture. So I went back to the conditioned media experiment. I had control cells, then a scrambled conditioned media, and again you see like a drop in proliferation and an increase in migration of the cancer cells. Whereas if I do the IFT88 knockout, it looks, the conditioning media experiment goes back to looking like the co-culture. So effectively, what we think is happening, I mean, I'll go to the whole working hypothesis now in a second, but basically it looks like whatever the osteocytes do to the cancer cells to stop them proliferating and make them more migratory, they seem to need their cilia to do it. And the cancer cells seem to be able to shut that down. So that sort of was as far as I got, but of course, yeah.
So I can understand this, interpret it. How are you culturing the osteocytes and then getting the conditioned media? So are these primary osteocytes? Are these?
So it's no. So there's all sorts of, so they're MLOY4 cell line. So if I was doing this with primary osteocytes, I mean, you wouldn't get enough osteocytes to do these experiments. Like, you need to use a cell line, and all of the cell lines have problems. They're all from mice, basically. So yes. Yeah, so they're 2D monolayer, yeah. And there's a problem because these are human cancer cells and it's a mouse osteocyte line. But I'll get on to, I did kind of try and overcome that later on. But I just sort of I basically had like dishes and wells everywhere, and I was like just trying to figure it out. No, so we did do mechanical stimulation. I'm leaving it out of the story because it complicates the story. But yes, mechanical stimulation interferes with all of this. And if the cancer cells don't have if the osteocytes don't have their primary cilia, they're less mechanoresponsive, so the effect that they have on the cancer cells drops off. But then I'm jumping ahead of myself, but in a chip model, we found that shear stress actually increased the invasion of the cancer cells. So I'm not sure if that is yeah, it's complicated, basically. So yeah, but I've left out, I was doing shear stress all the time, and it just complicates the story. So I'm sticking to the mechanistic one. So yes, so obviously the natural question was, what are the osteocytes doing to the cancer cells? What are they giving off? So this kind of led to a long kind of lots of cytokine arrays, lots of ELISAs, and I'll just cut straight to the chase and say that we looked at lots of different pro-inflammatory cytokines that could be coming off the cancer cell, the osteocytes. And the two conditions that we were comparing was, we wanted to see what changes in a similar way if you knock out the osteocyte's primary cilia via siRNA, or if you knock it out with TGF-beta, or if you knock it out with conditioned media from cancer cells. And we were looking for things that, I mean, it's not really showing well on this particular screen, but we sort of got like a heat map. We picked a few that changed really similarly and then didn't change much at all. So we looked at TNF-alpha, IL-6, IL-10, VEGF, and they were like the most strongly expressed changes. We added those to the cancer cells and then looked at their change in behavior. And unsurprisingly, they're pro-inflammatory. So we got increases in proliferation and increases in migration, but the only one where we saw a decrease in migration, a decrease in proliferation, but an increase in migration was TNF-alpha, and that was the only one that matched the original experiment, the original behavior that we were seeing. So nudging them towards more migration and slowing down their proliferation. So that made us think it might be TNF-alpha that the osteocytes are releasing into the media. And surely if that's the case, then if you go back to your conditioned media experiment at the very start and block TNF-alpha, then the conditioned media experiment should become similar to the co-culture experiment. And that's sort of what we saw. It's not a complete return, so there's probably more going on there, but we basically saw a similar drop, like a decrease in proliferation and an increase in migration that could be partially rescued by having a small molecule inhibitor of TNF-alpha in the media as well. So this is why I'm saying run away now, Fiona, because I'm not even showing like a fifth of the data. So anyway, so this is my working hypothesis that, in early metastasis, you've got your cancer cells, there's not that many of them. The osteocytes are signaling away and they're sending out TNF-alpha that seems to decrease the proliferation and increase the migration of the newly arrived cancer cells. I sort of think if this is happening, this might be why some cancer cells go dormant when they first arrive in, you can get a primary tumor and then, decades later have just a few cancer cells that remain in the bone marrow just explode back into life, and we don't know why. And I have a feeling that I don't have any evidence for this, but it might be to do with this. But basically, if the osteocytes are tamping down the proliferative behavior, then that might keep the cancer cells in line. But of course, cancer cells find a way around everything. So they would start growing, and if they're producing enough TGF-beta, there might come a tipping point where the TGF-beta can decrease the expression of the osteocytes' primary cilia, at which point you would shut down TNF-alpha signaling. And then you've got a vicious feedback loop in an established metastasis because you've got more proliferation, which leads to more TGF-beta, more osteocyte knockdown, cilia knockdown, and so on. And it would be exacerbated by the fact that these cancer cells also increase the osteoclastic behavior. And when osteoclasts dissolve bone matrix, they release TGF-beta that's latently sequestered within the bone matrix. So this would even further exacerbate this. So that's sort of where we got to with the paper. We then, at that point, I was sick of working with cells in a dish, and I think 2D monolayers aren't not real at all. Like, we're not flat, we're not 2D. So I was starting to move towards more organ on a chip type models. This is where I did get to look at shear stress, and we replicated some of the findings from before, but again, we were using mouse osteocytes. So we had an osteocyte channel. All stained cells here are stained in red, but the cancer cells all express EpCAM, whereas the osteocytes don't. So anything green is a cancer cell. And up on top, as Fiona explained, you've got two channels here. If you look at the fire diagram, and they overlap in the middle, and you've got a membrane that's got 7 micron pores, so the cancer cells can actually invade. And if all the cancer cells are if all the cancer cells are staying green in this Z projection here, which is kind of a Z focal stack down through the membrane, if you can see this kind of long kind of vague, maybe it's even better here, kind of a shaded area that runs along the middle, that's the membrane, so there's less fluorescence there. And what you can see is that if you don't have any shear stress stimulation of just the bone cells, this is a nice thing about organ chips, you can selectively stimulate different groups of cells in the same co-culture. If you get shear stress on the osteocytes, you actually encourage more invasion of the cancer cells, but not increased proliferation, which actually was sort of the opposite to what I saw in 2D monolayer culture. So yeah, basically, as soon as you move to 3D, everything gets more complicated, and often it gets counterintuitive as well. But I think is closer to what you see in real life. We're now trying to work to basically do away with cell lines as well as doing away with 2D culture. So more recently, what I've been doing is these are again kind of MLOY4s, and these are raw 264.7 osteoclasts, so still mouse cell lines. And what we've managed to do is suspend the osteocytes in a 3D collagen type 1 gel. They tend to maintain a dendritic-like morphology, but as we were talking about this morning, Tamara, they don't seem to form like a network, but they do slow down, thank God, because these things proliferate like crazy. So we're sort of getting, I'd actually be keen to try the OCY454 to see if they would work in there. But we also then have osteoclast co-culture with them, and they're sitting on a layer of DQ collagen. This is this collagen where if you cleave the collagen, it fluoresces green. So you can see where the osteocytes have been eating, basically. So osteoclasts, rather, have been eating. So if you look here, this is the osteoclast. It's got big, it's got multiple nuclei, which is what you want to see. And if you just look at the green staining, you can see basically a patch, a ring where the osteoclast is sitting. And that's because it's just been zapping away, using acid to eat away at the collagen. So it kind of gives you a measure of how much eating they've been doing. And we're now co-culturing them with osteoblasts as well on a top layer. But again, it's annoying me that these are all cell lines. So we're now starting to move, actually, sorry, I should say, we're trying to validate this at the moment. So we've got an animal model where there's 41 breast cancer cells have been injected. And this is on the left-hand side is like a section through it, and there's all stained positive with CK8 to identify the cancer cells, because the cancer cells express CK8, but the other cells don't. In the inset, then the purple staining is CK8 as well. So these are cancer cells crossing the membrane. See the way they've kind of funneled into kind of tight pores. That's them trying to cross and invade. So we're trying to find a way to compare the aggressiveness of the tumor inside the mouse. And try and get a measure that we could directly compare to what's happening in the chip. And we're also doing bulk RNA-seq on the resected tissue, and then going to compare the bulk RNA-seq on the cells within the chip, and hopefully see that at least it's somewhat matching the animal model. And we do have these animal models have been treated with chemo, so you've got control groups as well. So what we're hoping to see is that basically they'll match up nicely, and you could hopefully move away from using the animal models or use the chips to speed things up so you could test more quickly, because the chips take way less time to set up, and you can get more specific readouts. Of course, I want to move away entirely from cell lines, because they're just not, they're reliable and robust. Like, you can drop them from a building and they'll survive, but they're not patients, and they're not primary human cells. So we have replicated some of the findings that we had before with the TNF-alpha study using cancer spheroids, and we're co-cultured with the human MSCs, which we've nudged down the osteogenic pathway. They're not osteocytes, but I'd be keen to get help to try and make them more osteocytic. But they definitely are kind of osteoblastic. And then we're also working with the UK Biobank and the Barts Cancer Institute Biobank, because they collect tissue samples and then follow the patients on. So you have information on how the patients were treated, how long they live for, did they get bone mets, how long did it take them to get bone mets. So you could sort of try and see if the chip can replicate the patients. So that's where we're hoping to move next. Right, just to, now for something completely different. I showed you pictures at the start of kind of messed up osteocyte networks, and this is some of the work that I've been doing with Tamara. These are Tamara's amazing, and Charlie, they're amazing images from their aging study, because it's quite difficult it's very manual to try and measure that network. It's actually a pain, and it's a complete pain to measure the osteocyte network. But luckily, there's folks like us who love staring at these images all day long. And if you look at these images, on the left-hand side is the confocal images. If you look at the top left, you can see the osteocyte network in a young mouse, like a 2-month-old wild-type mouse. Right next to it, on the right-hand side, is the 36-month-old wild-type mouse. And if you just look, like even out of the corner of your eye, you can see that there's way more dark space. I mean, you guys would be familiar you've seen some of these images before, but there's just like, you can recognize the pattern there. There's less connectivity in that network. And then Tamara was also looking at TGF-beta receptor knockouts in the osteocytes, and basically found that the osteocytes have, if they've lost this receptor and can't sense TGF-beta, then they kind of, even though they're young, they've got almost like an old phenotype. And then I think partly because we were locked up during lockdown, Charlie did a fantastic connectomics network, which is a bit like, I kind of describe it like traffic on the highway. So if you do Google Maps and there's traffic on the route, you get a red mark like on the line on your route. This is kind of the same thing. So if you've got lots of options and there's not much traffic, and the network's really well connected, then it's all kind of stained in kind of green and blue. If it's red or yellow, then the osteocytes are not well connected, and if you were driving, there would be traffic because there's only a few routes to go. Okay, and what this analysis showed quite nicely was that the old mice had less connectivity. The network was less connected, and in the genetically modified mice, the same was true. And then I helped out a little bit by doing some models of the flow within the individual osteocytes. And what we found was that because the network is a bit messed up, the old mice and the knockout mice actually experience less shear stress. So not only is the network less well connected, it's actually less stimulated, it's less efficient. And what that sort of tells you is, it told us a lot, and it was really interesting. But to be honest, if there hadn't been a pandemic and we hadn't been locked up, I think I'd still be doing those models for you, Tamara. I never would have finished them because they take so long to run. And it kind of got me thinking that there just must be a way to speed this whole thing up, because at the end of the day, it's pattern recognition, and that's what machine learning is. So now what we've done is moved into using the same fantastic data set. We're looking at deep learning models. So we've combined computer vision with computer vision. You can think of it like, is it Waymo? Is that what you said the name of the cars are that go around and they just basically, all they're doing is, is taking lots of images really fast. And, is that a human? Last time I saw something like that, it was a human. So it's pattern recognition. It's the whole thing of AI taking over the world. It's just pattern recognition. It's not that big a deal. It just takes a lot of time to train these models. So we've used an attention unit and a convoluted neural network. And basically all it's doing is going from the scan I showed you before, it's thresholding it and then turning it into a network so the computer knows what it's looking at, and basically mapping out the individual connectivity of the network. And then once you do that, again, if you glanced at the image on the top left, and then glance at the one on the top right, you just as a human would know the top right is messed up, but the top left isn't. but the computer, because it's all been digitized now knows the difference. So this required like a lot of manual work and a lot of like picking individual pixels, but it basically about 120 hours. And the computer, once it was trained, was able to do it in about 10 or 30 seconds. So it just like, you know, basically that was 4 years of PhD worth of training for me, 120 hours, and I've basically been replaced. But that's the idea with AI, right? Is to replace yourself. And what was nice is that with a pretty high degree of confidence, so like 85% accuracy for the lacuna at least, it could distinguish between young and aged mice. So it saw very similar values, lower connectivity, lower dendrite length, lower numbers of osteocytes, and kind of a lot more dead ends in the aged mice compared to the young mice. Yeah.
Finish your thought, and then I'll ask.
Okay, I have like 2 more bullet points, and then I'm done. It could partially distinguish the TGF-beta knockout mice, but I think it's a more subtle change in those mice. So I think you'd need more training of the model before you could properly use it to dissect something like a genetic knockout. And then what I'm really hoping to do is, once we have it like better trained, then just apply it to a load of images from mice with cancer, because I know it will come back and say that's crazy, because I can tell by my eye it's crazy. So those are just kind of 2 projects the lab's been working on. I kind of wanted to give you a flavor of the in silico and in vitro work that we're doing. And yeah, I'm happy to answer any questions you might have, any of that. So thanks. Yeah, no, that's why I'm already done. And you should work with Fiona, because I did my PhD with her. So if take those positions, I would say, yeah, it was the best time I've had. So
There will be positions soon.
Yeah.
In your
Yeah.
So the thing I wanted to ask you about is this idea. I love what you've done with the AI version of analyzing the images. I mean, it's so attractive to think about that. All these different images that we have. So appreciating that this focus was on connectivity and looking at the extensions. How close are you with what you have set up to make this an AI way to look at, like, for example, knee joint sections, where you can binarize an image and just look at an intensity change and assign cutoffs? Like, how hard is it once you have this framework to adapt it to those nuances, or would that require starting from scratch?
So it would, if you applied, this is why I'm not scared of AI, because if you, let's say I have trained this so that it can literally, it would recognize every osteocyte network. If I showed it a picture of your lovely sections, it wouldn't know what to do, because it's not been trained on it. So you do need to start from scratch training a new model. If you want to do, you can do supervised or unsupervised learning. So if it's supervised learning, what you do is you basically tell it what you see. So you label the bits that you think are interesting, and then you tell it what those are. And then as it, learns, and you give it more and more examples, you can basically give it, whatever example you want, and it'll, as long as it's a picture of that, it'll know how close to normal it is, and it'll be able to measure it for you. So it's really not that hard. It's time consuming. If instead you want to look for patterns that you can't find or can't see, you can do unsupervised learning. So then you just give the computer tons and tons of images and let it look for patterns. But you get a lot of noise back with that. So you have to intervene at some point and say, you're telling me rubbish, you're telling me garbage. If you think about it, if you get asked to prove you're a human by Google, and you like find the traffic lights, like that, you're training those models. We've been doing it actually for about 20 years. And we haven't realized it.
So I think, like, we did a data set.
Yeah.
Here's all these images that we have. Here are numbers. Who cares what they are? Numbers indicating some outcome.
Yeah.
It can learn what those are by seeing examples, and then could it get to a point where it could assign them, or does it always have to be?
No, that's what it's doing. It's assigning them. It's labeling for. So when this image here, see, you can kind of see. So it's light gray where the lacuna are, and these are white. Like, that's because it knows that it's a lacuna. Now, if you found a very small blood vessel, for example, that was cutting through it has done that in the past, where it's gone, oh, cool lacuna, and you're like, no, idiot, that's a, God. But so you can get unlucky, and but what you want to do is, if you train it enough times, it will get a feel for, whatever. Like, I've been told that that's not a lacuna. Like, I get that it's a blood vessel, but it needs a lot of, really, you need to be in the hundreds and thousands of images before it can be as good as as guaranteed as good as a human. But they do learn very fast how to do an okay job. Like, within 100 images, you can get like a really, like, I think we're at 85% accuracy. We've only used about 100 images, which is mind blowing. Like, yeah. So yeah, no, it's totally doable. Yeah.
alongside all this training, did you also parameterize the images so that you can get connectivity junctions? And I'm thinking of all the terms that people use when they talk about trabecular architecture. So.
Yeah. So I used ones that we had used already for connectivity, dendrite length, dead ends. You can add more in, and the nice thing is you don't have to redo the training. So you just need to tell the model will have been trained. So all you need to do is tell the model, if you see something like this when you're being run, that's a dead end. So you don't need to go back and do all the training again. So the actual the manual training, you're really training the computer vision. Then the supervised learning is what happens later, and that's an easier ask. That doesn't take that long. So that the computer vision part needs a supercomputer, and you need access to high performance computing. The training can take a couple of days, but then once you have the model, it runs in 10 or 30 seconds. Yeah.
So of the original image, how much of manipulation of the thresholding and the resolution matters to then get to, I'm guessing, 2D of this for it to be able to pinpoint exactly?
Yeah, so we built in the thresholding into the model this time, and that's where the very first part of the paper, that it's a preprint now at the moment, is choosing the right thresholding method and the right thresholding parameters. Now, obviously, if you move to, what we haven't done is moved it to an entirely new data set, because then the thresholding might affect that, or the intensity of the stain might affect it. And that's why, really, if you want a general purpose model, it needs to be trained on different data sets taken by different people at different times. Because like Charlie is like an amazing, I mean, these are gorgeous images. But in reality, there'll be subtle changes depending on the microscope, depending on whatever, but it will get good enough. It'll recognize what noise is, because these stains in some sections had background. So there was kind of red stain coming through at the back where there was autofluorescence. And I figured in those areas it would really struggle, because it wouldn't, but it can tell. I think it's, the way the large language models predict the next word, I think it definitely sort of works in a way that it predicts the next pixel. So if it might see that the pixel next door is quite like maybe red enough to be a dendrite or a lacuna, but if it notices that there isn't one further on, that the path doesn't continue, it just won't label it. So because then it can't be a dendrite. If there's a kind of a clump over here of like light red, but it's not connected to anything, then it can't be a dendrite. So I
think you would also be able to measure the size of a dendrite in the different groups.
Yeah, well, so this is the trick. So I was labeling pixel by pixel. And what I'd love to do is see if you can measure PLR and the amount of expansion. But because of the resolution, I mean, the nanometer, you're on was 100 nanometers is the diameter, so you're close to the wavelength diffraction limit of light. So you get blur. But a pixel covers such a big chunk of that edge that if you click one extra pixel, you could be doubling the thickness almost of the space of the. So I think it would again need a lot more training before you could confidently say it's good at telling if that's a thick or thin. And at the moment, certainly we did. So he did. Simon did build it into the model, and he basically kind of it was just, there was no significant difference. It was just it just couldn't measure it basically, and so we just decided not to go any further. But I think if you trained it on enough images, and if you trained it using enough people, to kind of incorporate the variability and reduce the chances that you make a mistake, then yeah, I think it would train it. It would train that problem out, and you'd be able to measure it. But that's sort of a problem. It's sort of an artifact of the imaging as much as it is of the data.
I've got a last question.
Yeah.
Sorry. Your supervised detection, was he just you that were doing the imaging? Did you do a different person doing it? Would you see the same kind of?
So Simon was doing it as well, but he had a master's thesis to write up, a dissertation to write up. So, and I kind of was like, well, I'm the idiot who spent 4 years of my life doing this for my PhD. So I kind of I got my husband to do some as well, but that was, I mean, I don't know why I did that. But well, at least you got to see how painful it is. Yeah, you definitely need to get inter-operator variability in, partly because that makes the model more robust. It doesn't actually make it, even if, let's say you and I click completely different pixels, it just gets used to the fact that, oh, okay, this is what the general accepted, this is what more people agree on is a canaliculus. So yeah, it needs more of that needs to be built in. I'm kind of amazed that it really didn't take that much data to train the model to be this accurate. But having said that, I think if you wanted to be robust and general purpose, which I think is what we'd like to have like a model that people could just upload their scans or download the model and click run, and it would work for them and just be a free tool for everybody. I would want to be more confident that it would work in lots of cases, if that makes sense. Yeah.
Question about your culture work.
Yeah.
Between the osteocytes and so you've shown data for the MDA, so that's a triple negative breast cancer cell line, right? Have you seen any differences between the types of breast cancer and their crosstalk with osteocytes?
Yeah, slight. So that was a triple negative breast cancer cell line. I looked at MCF7s, which is an ER positive and probably more relevant cell line because it's most metastatic breast cancer tumors are actually estrogen receptor positive. And then I looked at 2 prostate cancer cell lines that were androgen receptor positive or negative. The trend kind of was the same, but the swings up and down changed. So I think it was less, the mechanism was just a bit less important for the MCF7s. And it seemed to be really important for one of the, like I saw bigger changes for one of the prostate cancer cell lines and then less changes. So it's variable. But then how good are the cancer cell lines? Yeah. So that's why I was like, rather than delving into it further and, I should just move straight to patients and see if any of this is relevant at all. So.
My second question was also very related to that. A lot of people do hypothesize this metastasis to bone that happens is because even as the tumor is progressing, you see the stiffness increasing at the primary site as well. And a lot of people hypothesize is that matching, it's going to a stiffer organ, it just helps cancer cells survive better, thrive better. And so do you think this loss of mechanosensing that happens in the osteocytes is kind of, I feel like cancer cells are the smartest cells in the body. I don't like that it's the way, but that's how it is. So are they just interacting with these osteocytes to downregulate their mechanosensing capability to create this perfect niche for themselves where it's just stiff enough where they can migrate, but not too stiff that it interferes?
Yeah, so it could totally be that. The reason I think that theory that it's stiffness driven has some that applies right to breast and prostate cancer. It doesn't work as well then when you look at other cancers because the tissues. Let's say if you look at like I can't think of an example off the top of my head, but if you let's say look. Multiple myeloma, right? Exactly. Actually breaks the rules altogether. So, but even if you had like a lung tumor that wants to go to the brain, like the brain is softer. But the lungs tumor is getting stiffer. So how come the cells maybe they want to run away from the stiff? So it's sort of, it's kind of, the trouble is the reason I think that those theories, even though people are working on them and they, like I like the theories, they give a lot of agency to the cancer cells. Like you just said, they are smart. But I don't know if they liked that it was getting stiffer, why wouldn't they just stay there? And also they have to basically become swimmers first, which is like the least stiff thing to do. So, yeah.
Part of the stiffness hypothesis is around the fact that cancer gets more aggressive. It's not like it's just like, oh, I need more beta podia out because my primary tumor site is now really stiff.
Yeah.
It's restricting me and I'm going to fight back.
Yeah. No, totally. I guess the challenge is that if it goes to, if it ends up in, the marrow is super soft and that's actually where it ends up first. It doesn't end up in the bone. And again, like it's flowing through, like it's under shear then for a huge chunk of its time going around the body. So, and actually it tends to, by the time we find the primary tumor, it's now known that the cells are already in our marrow, basically, like even if they might be killed, but they've made it to the marrow. So, and at that point, like you're only beginning to get like a stiff lump if you've just found. So I love the theory. I don't know how well it definitely works, if that makes sense. Yeah.
Along similar lines, so I was wondering about this crosstalk that you're sitting in osteocytes at early stage metastasis. And I guess one thing that I wonder is that is there a way to model this perhaps even like what it's still at the primary site? So if the cells haven't metastasized yet, but there's maybe some change that is systemically circulating.
Yeah.
That also influence whatever osteocytes are doing, or I don't know.
Yeah. Oh no, it totally could, and that's definitely what that's why Linda Bonewall's paper was so shocking, because it implies that whatever's causing the damage is actually systemic, and you don't actually need the cells there. So that was a bit wild. Because I'll tell you why I looked at metastasis. I kind of subscribed to what I was chatting to Val Weaver, who is here, and what she was saying was, well, she said a few years ago, she said students always come to me and they want to look at intravasation and extravasation and how these cells break away. And she's like, what's the point? They're already by the time a patient rolls into the clinic, you're dealing with the metastases, or there's already metastatic cells there. So it's too late. As an intellectual exercise, yeah, it's fundamental science, that's great. But if you want to be clinically relevant, it's the metastatic colonies that you really need to look at. So that's why I kind of stayed over there, to try and be more relevant. He says mixing things in dishes, But having said all that, I think it would be the great thing about the chips is you could hopefully get to a point where you've got something that's a bit like a tumor, and then at that point you could study how the cells kind of break off and travel through to another chip, and so have a multi-chip linked up, a bit like what Fiona was attempting to do. So yeah, it's worth studying, but I just stuck to the metastatic because I already had enough complexity on my hands.
With that, we'll thank our speakers again.
Thank you very much.